Hi, my name's Tom White, and today I'm going to be talking about running large-scale genomics pipelines on Dask and some of the lessons and challenges that we found when doing so. Okay, so to set the scene, I'd like to refer to this paper from about 6 years ago, which compares the data volumes and growth in those data volumes in various fields. In particular, there are two scientific fields here, and I think its conclusion was that the growth in genomic data was very large, and that's what we've been seeing. Indeed, the UK Biobank is a project that we've been using data from, and the scale of the data is what we've got on this slide, which is basically 10 million variants by 500,000 samples. So that's the size of the data array that we're processing. We've created a project called SGKit, which is a statistical genetics toolkit in Python. Why did we do that? Well, we wanted a high-quality PyData library for statistical and population genetics, and in particular we wanted it to scale down to be able to run on a single machine, but also to scale out to run on a cluster of machines. And we thought that the existing solutions for that did not achieve those goals, particularly in a Python context. The people behind it are from the Oxford Big Data Institute, who are also behind projects such as scikit-allele and tskit in similar areas, and also the company called Related Sciences. So SGKit uses three main PyData libraries. They are Xarray, Dask, and Zar. Xarray is used for its metadata representation of arrays and datasets. Dask is used for computation, and Zar is used for storage, in particular cloud storage. This diagram shows the layout of the data. There's quite a bit of detail on here, but the main idea is that there are two dimensions of interest. Variants and samples, which I mentioned a moment ago in the context of the UK Biobank. Variants are to do with the position on the genome, and samples are to do with the individuals, so individuals giving their DNA samples. And the cells in this array correspond to what that particular individual's DNA is at that position in the genome. In terms of what it looks like from a kind of coding point of view, this is a very small example which shows a simulated data set with 1,000 variants and 250 samples, and the bit at the bottom shows the variables in Xarray. So you can see the different call genotypes and various other variables. And this is just a regular Xarray, which can be manipulated in using regular Xarray functions. What SGKit does is create some methods that are genomic methods for manipulating genomic data in ways that geneticists are familiar with. So to talk about the Dask challenges that we faced, there were 5, which I'm going to summarize now, go through now. The first is representing missing data for ints. Genomics is interesting because it uses small integers, typically 0, 1, and missing. So we don't particularly want to use floats with nans to represent missing, because it uses at least 4 times as much memory as is needed. So we thought about using masked arrays, but unfortunately the support in the various backends that we want to use is not uniform. In particular, CuPy does not support masked arrays. So, and there's the issue for it. And so as a workaround, we use a sentinel value. So we represent missing as -1, but that means that we have to have special casing in, for example, our number routines. So basically, we would like, masked array support in CuPy, or possibly even a float array, a float 8 type in all of the libraries. that would be equivalent, I guess. The second challenge is optimizing Dask chunking for performance. Getting the right chunking is critical to performance. We found that it's very sensitive, so you can change the chunking in a particular input array and the performance will change dramatically. So here's an example of a 4x speed up that we found. Unfortunately, you have to manually examine the shape of every intermediate array in a computation, and that's pretty labor intensive, and it's also often quite counterintuitive. So you think that if you change it in a particular way it might improve things, but in fact it often does the opposite to what you expect. It's also unpredictable. The actual Dask rechunking operation is unpredictable in terms of time and memory, and we've had to use the rechunker library in several cases in our code. So I think what we would like is better tools for choosing a chunking for arrays, and possibly also to incorporate some of the ideas from the rechunker library into Dask itself. The third challenge is scaling linear algebra operations. We found and fixed quite a few matrix multiplication and SVD bugs in Dask. Here's a list of them in case anyone's interested. So I think the kind of takeaway is that Dask works very well for these operations, but sometimes there are corner cases where you have to kind of take care, and there may be improvements that are still need making to Dask itself, which is fine. And we think though that it would be very useful to have some scalable linear benchmarks, linear algebra benchmarks for Dask to keep an eye on its performance for different operations. Slab is a project which does something like this, which might be interesting. Fourth challenge is kind of related to the previous two. There's an overwhelming amount of detail at the task level when you're trying to debug what's happening to a task graph. So typically what we've done is downsamples to a subset of the input data to manually see what's going on in the computation. And that can be quite tricky. You have to be quite careful, and it depends on the computation that's being run. So what we'd like here really is a high-level visualization of computation, and I think that's coming in Dask. There's a lot more work going on in the high-level graph code, and so we're hopeful that this is going to be improved. The final challenge that we've had is choosing a stable Dask release. There are lots of Dask releases coming out. It has a pretty frequent release tempo, which is a good thing. But we found that some versions or combinations of Dask and Dask distributed were unusable. And there's a reference. But the problem is it doesn't always manifest itself when you're running tests at a small scale. It's only when you start running on a cluster and you start to really exercise Dask that we start to see the errors. So we like the idea of having blessed releases where you can be more sure that a particular release works well at scale. And there's discussion about that on a community issue. So yeah, we think that there's a lot of scope for improving that, and hopefully the vendors would step in and help with the quality testing now. So that's it from me. Thank you very much.